Hello, everyone. Welcome to the second podcast over Chapter 18 for Day 19. This will be the last podcast for this day. In this podcast, let's back up. In the last podcast, we talked about how genes are regulated and focused on how bacteria regulate their genes. And in this chapter, in this objective, we're going to focus on how eukaryotic cells regulate their genes. So let's go ahead and get started here. So, as we begin this discussion of eukaryotic gene regulation, I think it's important for us to go back and redraw our cell that we had on the first one. And remember, we had our signal that bound here that told the cell within the nucleus, let's draw our nucleus here, which genes to turn on. Okay. And within our nucleus, we had our chromosome here. So let's just draw the DNA. And then that this was transcribed, TXN for our abbreviation, to make our messenger RNA. And then it gets processed, and because of space, I'm just going to put a P here, but in your notes you should write process because it gets processed to give us our mature messenger RNA. And then this mature messenger RNA, remember with the G cap on it, the spliced out introns and the poly A tail, now we'll leave the friendly confines of the nucleus and go into the cytoplasm. And let's go and draw this out with our G cap, the message itself with the poly A tail. Then this gets translated, translation or abbreviation, and we get our protein. And then our protein is modified. It's folded, fold and modified. So fold and modify to give us our mature protein. And in the final act of regulation, this mature protein can be degraded. All right, so all of these things happen in the cell to regulate a gene. So let's talk about numbering them again. So here, the signaling right here. The packing of the chromosome, that's another level. Transcription is another level. Processing is another level of regulation. Export is another level. Translation is another level, so that's 6. Modifying folding, that's our 7th. And then degrading is our last one right here. In this podcast, we're going to spend a lot of time, I'm going to put a star here, talking about how chromosome structure can affect gene transcription. So let's put a star here because we're going to spend a gruesome amount of time on that. And transcription, we're going to spend a lot of time on that too. I want to spend a little bit more time talking about processing. It's more of a reminder, so I'm just going to put a dot here because I want to spend some time on this page talking about it. And I also want to spend a little bit of time talking about the messenger RNA because we didn't spend much time about it, but how the structure of this messenger RNA depends upon what will help determine how that gene is regulated. And those are the main areas we want to talk about. So I'm going to say a little bit about this messenger RNA, its structure, how it determines how long this gene will be expressed, and the process of the mRNA. And then the last 2 slides, we're going to talk a lot about the chromatin and transcription. So before we move on, let's come over here in a little area here and talk about our messenger RNA. Just to remind you, we've had this before, but remember this messenger RNA has our G cap on it, and we have our messenger RNA with our poly A tail. And remember, we have exons and introns. E1, I, E2, I, and E3. All right, remember we said because of alternative splicing, we could make 2 mature messenger RNAs from this. We could make one that has E1 and E2 and E3. Or we can make one that just had E1 and E3. They would still have the G cap and the poly A tail. I'm just not drawing them right now. So this is another very important level of regulation, determining which introns are going to be cut out and if any exons are going to be cut out as well is an essential step in regulating this gene. The next thing I want to talk about is the length of the poly A tail. And so let's just draw two genes here, two messenger RNAs. And this one, let's put just three, well, we'll put five adenines. And this one here, we're going to put 5, but then I'm going to make a nomenclature saying we're going to repeat this 10 times. So we really have 50 adenines here. And the point I want to make here is that the longer this poly A tail, the longer this messenger RNA will survive in the cytoplasm, because as these messenger RNAs are degraded, they're degraded at the 3' from the 3' end. So this poly A tail protects it until you start getting into the critical part of the message. And once this poly A tail is gone, the rest of the messenger RNA is rapidly degraded. So again, the longer the poly A tail, the longer that messenger RNA will be inside the cytoplasm. In addition, once that poly A tail is removed, it can't be translated anymore because the poly A tail is important in binding to the ribosomes to initiate translation. So again, the longer this poly A tail, the longer it survives. Now, on the next slide, I want to focus in on step 2 here. That is how the chromosome is packaged. So we said we're going to talk about how genes are regulated at the level of the chromatin structure. If you remember a while back, we talked about how chromosomes were packaged together with DNA and these special proteins called histones. So what I want to do here is I'm going to draw a piece of DNA. I'm going to draw a piece of DNA here. And first, let's just draw it completely open like this. Okay, this is a double helix. I'm just not drawing the double helix. This is a piece of DNA. And we know that this is packaged using these histone proteins that we talked about. So it looks something like this. Okay, let's just be kind of generic in our drawing here. But it's packaged up nice and tightly like so, something like that. And that's because these histones are these, let's just make them blue here. What they do is they wrap around, or rather the DNA wraps around the histones to hold it together in this really compacted structure. Now the problem comes when we want to turn a gene on. So let's draw a gene, and let's put our gene here in our fully unwrapped DNA. So here's our gene, and let's put our promoter here. If we wanted to turn this gene on in this folded out piece of DNA, we could easily have our RNA polymerase bind here to the promoter and transcribe this gene, and then would make the protein. Now the problem though is our gene now is wrapped up into this packed up DNA with histones on it. And so I'm going to put it right here. So it's all packaged in here. And so the problem comes now when our RNA polymerase, if I called this DNA polymerase, I should have called it RNA polymerase just in case I made a mistake. This is RNA polymerase. When it tries to gain access to this gene's promoter to activate transcription, it can't. It just can't find the promoter, and it bounces off, and it goes to try to do something else. So in order to activate this gene, we have to unpackage this chromosome. And we do this in an interesting way. We add chemicals to these histones. The chemical is called an acetyl group. We're not going to ask you acetyl on the test, but I'm just going to tell you what it is, just in case you're ever on Jeopardy and they ask you that question, you'll be able to answer it. You'll also need to know this as you move forward, so we might as well at least use the right terms now. But on a test, we are never going to use the word acetyl. And if we do, you can pull this podcast up and all right, so it adds these acetyl groups on it. And when we add these acetyl groups on it, what happens is something rather magical. Now, we're only going to do it to this gene, so what we're going to have here is we're still going to have some of our chromosome packaged up with the histones like so. We still have histones associated with regions of the DNA that are not supposed to be expressed. So some of it's still packaged up nicely like so. But now what happens is our gene is exposed. So here's our gene with the promoter there. And now our RNA polymerase can have access to this promoter, binds to the promoter, and can turn on transcription. And when we do that, up here I should draw the These histone proteins, they've fallen off, remember. Sorry, the size is not proportional here to what I've already drawn. And they still have this chemical on it, this acetyl group. Again, don't worry too much about it for the exam, but you'll need to know it later. You'll also learn the enzymes that do this, and there's so much more fascinating things you're going to learn about this. So they fall off, and as long as these acetyl groups are attached to the histones, they're not going to be able to bind back here. So as long as they're off this gene, the RNA polymerase will bind to the promoter and make messenger RNA from this gene. This is an incredibly efficient way, or common way, I should say, that eukaryotes from plants to animals to fungi will turn on genes by modifying the chromatin. In fact, we should call this chromatin structure modification. It's probably the best way to call this. So let's just review this real quick. Our chromosomes are packaged up, and if this is, sometimes I'll interchange the word chromosome and chromatin. Remember, the distinction is chromosomes are found in mitosis, chromatin is found during interphase. And so really, we should call this chromatin because gene expression happens during interphase. Okay, so our chromatin structure here is packaged up with these histones. As it's packaged, any genes within this fully folded up packaged chromosomes are not accessible by the RNA polymerase. To gain access, small little chemicals have to bind to the histones. When they do that, the histones change shape and they fall off. When they fall off, the DNA unfurls, it comes apart, and now RNA polymerase can bind to the promoter and activate transcription, and then we get our messenger RNA with, the G cap and the poly A tail and all that good stuff. Okay, this is what you need to know for chromatin structure modification. Okay, let's talk about gene regulation at the level of transcription. Okay, so we have our gene, and it's just been released from the chromosome, so to speak, unpackaged because of the histones falling off. And so, if we want to, we could draw what we had on the last slide with this part of the chromatin still being tightly packaged, and our gene of interest that we want to activate right here. And we can still draw our histones on this just for the fun of it. So our histones are still packaging up this part. But let's not worry about that. Let's focus on this area here, because this is where transcription is going to happen. We're going to finally be able to activate this gene and make the protein that we need to have that desired response the cell wants to have. So over here, this is going to be our gene, the reading frame of the gene, the part that's going to make the messenger RNA and go on to make the protein. Just upstream of this, we have our promoter. So I'm just going to write PROM here. That's our promoter. Now, up to this point, we've always just told you that the RNA polymerase binds to this and we have transcription. Well, we left out some details. So let's draw our friend, the RNA polymerase, right here. And we're still going to leave out some of the details. This is another story that's going to build upon as you move through the curriculum. So how does the RNA polymerase bind to the promoter to activate it? Well, it's going to do this by using several things called transcription factors. And just like we saw before, we have this Tata box here. We have our Tata box here. We'll make it in blue, or I guess red. And so how does the RNA polymerase know to come here? Well, what happens is we have these other proteins called transcription factors. Transcription factors, in fact, to turn this gene on, we need roughly 20, in some cases 30 or more transcription factors to be involved here. And transcription factors are just proteins. So we're going to have this transcription factor bind here. In fact, it's the first one that binds. It's got a very convenient name. It's called the Tata binding protein. Okay. I suspect you can guess what it does. So I'm going to write TATA-BP for Tata binding protein. It is what binds to the Tata box. And now the RNA polymerase can bind to this transcription factor. So now I'm just going to draw the RNA polymerase kind of on top of this one. And now that it's bound, in theory, it can now move forward and transcribe this gene. I say in theory because really to get this going, we need more transcription factors. And these transcription factors, they're going to bind to different parts of the RNA polymerase. They're also going to bind different parts of the DNA. So I'm just going to kind of draw them around here. It requires several proteins to bind to this to make it go. And this is really important because once you turn this gene on, there's no going back, right? Now you've transcribed it. So we want to be careful. There's several lines of regulation in eukaryotic systems because they're a little bit more, they're in some cases a lot more complex, and so regulating which genes get turned on is a much more elaborate show. So you should know this much so far, that the RNA polymerase binds to transcription factors that are bound to the promoter so that it can transcribe the gene. So you should know, let's make a list of things you should know regarding gene transcription at the transcription level. You should know transcription factors. I'll use my abbreviation TXN, factors. These are what decide which gene gets turned on. So they're the master regulators of this. We also need our RNA polymerase. We should also remember we need the promoter with the top box. So I'm going to write promoter here. These are all important. And then there's one more thing we've left off that we need to make sure we talk about. And that term is called enhancers. And as the name implies, they kind of enhance this process. They add this extra level of sophistication to turning these genes on. And they're kind of interesting because everything we've talked about up to this point, all the proteins, all the transcription factors, the RNA polymerase, are localized right next to the gene that needs to be turned on. Enhancers, they can be located very far away. So let's extend this chromosome out some. They can be localized way over here. So we have enhancer DNA sequences here that enhancer proteins bind to. And so how does this protein far away, thousands of base pairs, tens of thousands of base pairs away from this promoter affect the transcription of this gene? It does so because our DNA, our chromosomes, are very flexible. And what happens is that this bends, and as it bends, it will bring the DNA and enhancer right here. And so now, if we can make an attempt at some kind of time-lapse photography, the DNA and the enhancer move up here. So now we have our DNA wrapping around here with our enhancer right here, our enhancer protein right here. And we're just going to get rid of remember, this isn't here anymore. So remember, this, of course, isn't here anymore. It's been moved up here, okay, with enhancer, and it brings enhancer up there. In addition to the enhancer proteins coming up here, there are other protein transcription factors that now come along with it. Okay, I'm not particularly proud of this drawing. I could have done a lot better, I think. But in the interest of time, we're going to let this go. All right, so we need these things to happen in order to turn on a gene at the level of transcription. We need our promoter in front of the gene of interest. The TATA binding protein, one of the transcription factors, binds to the promoter. When it binds to the promoter, it will recruit RNA polymerase. Once RNA polymerase is bound to that transcription factor, it's now poised to transcribe this gene. What makes it possible to do that is additional transcription factors will bind. In addition to that, these enhancer proteins that are located far, far, far away are brought close to it. And once they touch this area here, that fires this RNA polymerase to move along here and make our transcript. So we need all four of these things to happen to turn on this gene. Okay, well, I felt bad about that drawing I made of eukaryotic gene regulation at the level of transcription. So I found this video that shows it pretty nicely. So let's go over it again, and I'm going to highlight the parts that we talked about when I went over the drawing myself. So let's look at this real quick. Here we have the gene in yellow, and then our promoter here. This promoter, I'm going to hit pause here, has that region known as the TATA box. And these orange proteins coming in, remember I said you might have up to 20 or more proteins coming into this region to initiate transcription. They're going to bind to this promoter, promoter at the TATA box region. And now they're going to recruit RNA polymerase. It's going to come in here, right there, and it binds to these transcription factors, and it also binds to the DNA. We're also going to see additional transcription factors come in. They're going to call them something else, but for our purposes, we can just call them transcription factors. And as soon as it binds, see what happens here? It's going to release the RNA polymerase. And now it takes off and makes the messenger RNA. Now, one thing that I tried to draw, it was really kind of hard to draw in a one-dimensional screen here, were these regions right here that are far away. They could be, remember we said they could be thousands of base pairs away. And we call them enhancer sequences. And this enhancer protein here is going to bind to it. So let's watch what happens here. It binds to this enhancer region on the DNA, and now this bends the DNA, and now it can exert its positive regulation here, and the RNA polymerase will now make the transcript. Okay, I'm going to stop it there. We didn't talk about repressors here, so I'm not going to bring that up now. Okay, if you have any questions, make sure you come see us, and if not, we'll see you in class. Bye.